Hello. Today in this section, we'll be walking through how to properly normalize a protein of interest using a validated housekeeping protein. Before using housekeeping proteins in a normalization strategy, such as this, you must always keep in mind that you need to validate the housekeeping protein first. Many journals now require a validation if you plan to use housekeeping proteins to ensure the experimental conditions you're using do not affect the housekeeping protein and thus ultimately your data. If the validity of your housekeeping protein is in question, or if the housekeeping protein you're planning to use or are using is known to be affected in your biological system, then it's best to use an alternative method such as revert total protein stain. For the rationale behind using this type of strategy, and for more information on selecting a strategy, it would be best to review the earlier lessons in this course. Here I have the result of a Western blot experiment loaded into Image Studio Lite software. In this experiment, 4 samples of interest were run in triplicate across a gel. After running the gel and transferring to a membrane, a Western blot was performed to detect actin in the 700 channel, shown here in red, and then to multiplex at the same time, an antibody directed against ERK was detected in the 800 channel, shown here in green. This Western blot was imaged on an Odyssey imager and then imported into Image Studio Lite. After importing, we've already gone ahead and gone through and quantified our bands of interest. Now, to recap the basic features of how to quantify your targets of interest, especially in Image Studio Lite, please make sure to review the lessons covering basic quantification. Now, once you've gone ahead and quantified in Image Studio Lite, we'll next want to go ahead and export our data to a graphing program. Here, what I've done is I've taken the data from Image Studio Lite, and I've gone ahead and imported it into Excel. In Excel, the quantification from the actin is placed into one tab, and then the quantification from ERK into a second. In our normalization analysis, our first step will be to calculate a normalization factor, which will be used to correct the calculated signal values for our ERK target. To do this, we'll first want to examine the calculated signals from our actin antibody, shown here. As we examine the values, we're going to want to go ahead and look from lane to lane, and then find the lane with the highest signal value. As we do that here, we can see this lane here in lane 11 has the highest value. Once we've located this value, we'll next want to go ahead and divide the other lanes by it. This can be easily done in Excel by going ahead and in a cell, dividing the adjacent cell by that value, in my case, 5440. Once we go ahead and hit enter, we've now determined the normalization factor for that lane, and then using Excel copy and paste functionality, we can go ahead and apply that same formula to all lanes. This calculated value is our normalization factor, which we can type into the table for easy organization. Now that we have this value, we can go ahead and select all of our values, and then copy it, and then paste it into our ERK table. Now, when you paste it into your ERK table, make sure you paste the values and not the formulas. Now we will go ahead and use this normalization factor to determine what our normalized signal is. We will do that by going ahead and dividing the signal value by this normalization factor. And this will produce a normalized signal using that factor. We can now go ahead and use copy and paste functionality to determine the normalized signal for all lanes. Now at this point, we've successfully gone ahead and used our signals from our housekeeping protein actin to calculate a normalization factor. We then use that normalization factor to correct our target of interest, ERK. Now in this specific example, I've taken the same sample and run it in triplicate in a Western blot. So what we can go ahead and do is use Excel to go ahead and calculate those averages. So in my experiment, I have an average of lanes 1, 5, and 10 as one sample. I then have lanes 2, 6, and 10 as my next. For my third set, I have lanes 3, 7, and 11. And then my final set would be the averages of lanes 4, 8, and 12. Now we can also use Excel to go ahead and also plot the standard deviation of each of these as well. So now we've used Excel to go ahead and generate the averages of my 4 samples and the standard deviation of each. If I want, I can also go ahead and select this data and using even more Excel functions, create side-by-side plots of this data. Since I have the standard deviation, if I want, I can also add additional elements encompassing that standard deviation as well into Microsoft Excel. Now, as you work forward and continue to analyze these samples or do other treatments, just make sure that you always use this normalized signal as it's taking the housekeeping protein into account to correct for any variation from lane to lane throughout the experiment. Those are the basics of how to accurately and precisely normalize a target of interest using a validated housekeeping protein in Image Studio Lite and Microsoft Excel. If you want to learn more about Image Studio Lite, please visit www.licor.com/islite. Thanks for taking the time to watch these lessons, and good luck with your own analysis.